All right, this is going to be transcription translation practice. So what's always given to you is DNA. So DNA to mRNA is going to be transcription. The very first thing that you need to know how to do is figure out your mRNA, transcription. So we have 3 different ways that we can do this, and I'm just writing them out. So DNA is going to be unzipped. You're only going to copy one side and you're going to create mRNA. So mRNA is always read in 3 codons. I have to write these small. So we are doing transcription. All right, for transcription, you have to find the complementary base pair for mRNA. So remember, for RNA, you should never, ever have any T's. All the T's that would bind with adenine are going to be replaced with U's. So TAC, the opposite, is going to be AUG. The next one, GCC. The next one, AGC. The opposite of A is UAA. So that would represent transcription. This is practice. Transcription is going from DNA to mRNA. Now, in order to go from mRNA to protein, which is going to be translation, you need to understand the genetic code. So translation is going to be using mRNA to figure out your protein. A protein is basically an amino acid sequence. So you use the genetic code. This is one example of how the genetic code would look. You can see that there are U's inside of the genetic code, which means this is from mRNA. The genetic code is the 3 codon sequences from mRNA that are going to code for amino acids. So these are codes and these are amino acids. So when we do the first one, AUG, if you're using something that looks like this, the first letter is A, the second letter is U, and then the third letter is G. So the first codon is called AUG, and the amino acid is methionine. Now, methionine is always the first amino acid to start translation. So this would be MET to represent the start amino acid, and you circle it. Now, translation occurs at the ribosome, which builds proteins, and proteins are amino acid sequence, and it's going to place peptide bonds in between. But first we're going to translate it. So we read the next one, GCC. You go to the first letter, G, CC is the codon, which is going to be for ALA, which is alanine, which is an amino acid. So you write ALA, and that is the next amino acid. Now, mRNA, AGC, you find your first letter, A, G, C, amino acid. The AGC encodes for is serine. Serine is the next amino acid, SER. It's an amino acid. So then you do UAA. Well, UAA is known as a stop codon. So you literally write stop. If you do translation correctly, you should have amino acids with the start amino acid, which is methionine, and you should end with a stop codon, which ends translation. So anytime I give you something, I try to give it to you so that methionine is first. So when you're doing your protein or your amino acid sequence, this is your start amino acid. And the stop codon, UAA, is going to tell us to stop translation, and these would be your peptide bonds. So when you're doing translation, you have to use the genetic code. The genetic code is going to be a chart that looks something like this. Some of them that I've seen is circular, and it's got the codons inside, and it tells you the amino acids inside of it. So there are two practices that you can use here. So you can push pause, and you can see if you can figure these out. So I've figured out all of my codons for mRNA. Always double check to make sure that you do not have any T's. Now, you're going to use the genetic code to figure out your amino acid sequence, which is going to form your protein. This is my amino acid sequence. Again, proteins, our amino acid sequence, peptide bonds are going to be found in between. It always starts with a start amino acid, and it always ends with stop because we have stop codons and start codons. So AUG always represents your codon, your start code. UGA is a stop codon. UAA is a stop codon. AUG is the start codon. AUG is the start codon. UAG is the stop codon. So this is practice on how to use the genetic code.